My name is Maria Winkvist and I'm working at JoVE Healthcare in Uppsala in Sweden. Cell surface proteins are often involved in communication between cells and their environment and are therefore of particular interest in research aiming to gain more insight to diseases as well as other biological variations induced by changes in the cell environment. In this video, I'm going to show you a simple CyDye DIGE cell surface labeling protocol, and you will be able to visually enrich these low abundant proteins without physical enhancement and study differences of these cell surface proteins with the Ettan DIGE technology. If you find this technology interesting, you can find more information in the 2D handbook. Moreover, we have an application note on this particular topic, which you can download from JoVE.
In the cell surface labeling protocol, seen on top, you label the cells while they are still intact. During the labeling process, the dye will only have access to the cell surface proteins. After the labeling step, the cells are lysed. To verify cell surface specific labeling, the labeled sample was fractionated into membrane and cytosolic proteins. A non-fractionated sample was prepared in parallel for comparison. This fractionation analysis is not necessary, but was done here just to show that the cell surface protocol is specific for cell surface proteins. We also performed the standard Ettan